can observe one thing. These two functional groups or atoms which are attached to the double bonded carbon-carbon, then what will happen? This bond will twist. And when there is a twisting, what will happen? There is this bond between these two carbons will tend to break. Is not it? So what we have to say? We have to say that the way the single bonded carbon or the atoms which are attached to the single bonded carbon atoms, they can be rotated freely about the carbon-carbon single bond. In the similar way, we cannot rotate the atoms or functional groups attached to a doubly bonded carbon-carbon bond that easily. And from this, the term restricted rotation comes into preview. Okay, and to make this or easily or to make the things simpler, we have taken the example like this. Okay, here you can see there are two plies which are attached to each other through two nails. Let us assume that these two nails, they are representing the double bond of an alkene molecule. Okay, whereas in this case, let us assume this nail which is joining the two pieces of plies as a representation of a carbon-carbon single bond. Now here we can observe one ply can be freely rotated about this nail or the so-called representation of carbon-carbon single bond. However, if we want to rotate one of these two plies, or if at all we want to rotate both of them, is it possible? It is not possible. Why? Because there are two bonds which makes the rotation restricted, or which restrict the free rotation about the double bond. Isn't it? As a result, the term restricted rotation or restriction in rotation is clear. Is not it? Now let us consider the case of 2-butene. Here we can see it exists in two spatial arrangements. When we talk about 2-butene, what are the two spatial arrangements? Here we can see carbon-carbon double bond, which is the basic feature of an alkene, followed by, as because it is 2-butene, the double bond is at its position number 2 and it has got 4 carbon chain. First carbon, second carbon, third carbon and fourth carbon. This is representing one orientation or one spatial arrangement, and the second spatial arrangement, here you can see first carbon, second carbon, carbon-carbon double bond at position number 2, third carbon and fourth carbon. Now here, what is the difference? Here we can observe both the CH3 groups attached to the carbon-carbon double bond, they lie on the same side. Whereas here, if we observe clearly, we can see that the two CH3 groups, they lie on opposite sides. Okay. So we will call such type of compounds as cis compounds. Here, as because the structure is 2-butene, we will add the spatial arrangement or spatial description. We will add the description of spatial arrangement of 2-butene by adding the term cis 2-butene. Cis 2-butene means we want to specify that in 2-butene, both the CH3 functional groups are placed on same side of the double bond. Here, we can specify this type of spatial arrangement by using the term trans. Hence, this structure can also be called as trans 2-butene. Okay, if we call it as 2-butene, we are not giving any idea regarding the spatial arrangement of the atoms or functional groups that are present in the molecule. So when we add the term cis or trans, we give clear idea regarding the spatial arrangement of the molecule. Clear? When we are talking about geometrical isomerism, it is important, or it is very important, to specify the exact orientation of the molecule in space. Okay, so we can say the two compounds here are referred to as cis-trans isomers, or we can say these are the geometrical isomers of 2-butene. And how they are distinguished? They are distinguished from each other by the term cis and trans. The cis isomer is one in which two similar groups are on the same side of the double bond. Whereas in case of trans isomer, what happens? Two similar groups, they lie on the opposite side of the double bond. So we can conclude this geometrical isomerism can also be called as cis-trans isomerism. Now let us see this cis-trans isomerism or cis-trans geometrical isomerism in detail. So when we talk about alkenes, alkenes can be stereoisomeric, even though this is slightly different than what we learned about enantiomers and diastereomers containing chirality center. Now, when we talk about this chirality center, we talk about asymmetric carbon. Asymmetric carbon is one which has got 4 different functional groups attached to it. Now here we are talking about alkenes, cis-trans alkenes. So here there is no question of this chirality. That is because the carbon atom here is associated with the double bond. Is not it? So for better understanding, let us take some examples so that the concept can be more clear. Here, let us try to determine the relationship between molecules in each of the following pairs. Now here, if we observe this first diagram, here we can see there are 2 images. Okay, this is a structure where the chloro substitution is placed towards the observer, whereas here in this molecule we can say, as because it is represented by a base bond, we can say chlorine atom lies away from the observer. Okay, here we can say both are having mirror image relationship with each other. Now let us see these 2 structures. Here what will happen, here what is observed? Here if we see, the first chlorine atom is attached to this carbon atom in such a way that the chlorine lies away from the observer, whereas here the structure shows that chlorine is placed towards the observer. Now, if we compare this structure with this structure, here we can observe both the chlorine atoms are attached to this chiral centers in such a way that both the chlorines, they lie towards the observer. So in short, we can say these two structures, they are not representing mirror image relationship with each other. क्या ये दोनों एक दूसरे के mirror image हो सकते हैं? No. Now let us take an alkene in this category, for example. Now here we can observe in both these structures, we could see chiral centers. However, here when we talk about these two structures, here we can see there is existence of a double bond in the molecule. Okay, when there is double bond, there is a case of restricted rotation, isn't it? Here we can say the first two molecules are a pair of enantiomers since they are non-superimposable mirror images of each other. Now, when we talk about these two structures, they are also representing the same molecular formula. However, they are representing diastereomers because they are stereoisomers, but they are not mirror images of each other. So we can summarize stereoisomerism can be broadly categorized into two parts, that is enantiomers and diastereomers. Enantiomers, they bear mirror image relationship with each other, whereas diastereomers, they are not mirror images of each other. Now, what is the relationship between the two alkenes then? It is clearly not identical. Now, when we talk about the two alkenes, they are not identical. Here we can see this is the double bond, and to this doubly bonded carbon-carbon bond, here one ethyl group is attached, here one ethyl group is attached. Here one attachment is towards the top, whereas second attachment is this ethyl group is lying below the plane. Here we can see both the ethyl groups are lying above the plane, because whenever we talk about double bonds, we talk about sp2 hybridization, and sp2 hybridization always represents a planar structure, isn't it? So to conclude, we can say in case of these two alkenes, they are not identical because the atoms on the doubly bonded carbon-carbon are pointing at different directions. They are also not constitutional isomers. Can we call them as constitutional isomers? No. Since all the atoms are connected in the similar way, there is no change in connection of atoms. Sequence of attachment of atoms in the molecule remains the same. However, what is different between the two alkene molecules? In this example 3, the way they are attached or their orientation in space. That is the only difference. So they cannot be called as constitutional isomers. Okay. They are not mirror images also. Hence, they are classified as diastereomers since they are representing stereoisomers. Okay. Here we can see the same example. The two ethyl groups, they are lying on two different sides of the planar bond. Here we can see both the functional groups, they are similar and they are lying on same side of this planar bond. They are stereoisomers, but they are not mirror images of each other. Hence, they fit perfectly to the definition of diastereomers. In order to distinguish this specific type of diastereomers, in case of chiral molecules, They were classified in a different way. However, when we talk about alkenes, these diastereomers, they can be classified as cis and trans isomers. Now, how this cis-trans isomerism is determined? Let us see that. Here we can, the first thing that we have to do is we have to first locate the position of the double bond. Then we have to see what are the various atoms or functional groups that are attached to both the doubly bonded carbon atoms. Here there is one carbon, here there is second carbon, and these two carbons are attached to each other through a double bond. Okay. Now, if we consider this as the plane of separation between these two doubly bonded carbon atoms, then what we can observe, we can see the ethyl group which is attached to carbon 1 is lying here, and the ethyl group which is attached to carbon 2 is oriented in this way. Now, what is the difference? Both are ethyl groups. However, what is the difference? Here we can see both the ethyl groups, they are lying opposite to each other. If these groups, they lie on the same side of the double bond, then we call the diastereomer as cis isomer, and if they lie on opposite sides of the double bond, then we will call it as a trans isomer. Now let us see this cis-trans isomerism in detail. Here we have taken the example of 3-hexene, the same molecule. Okay. See the orientation of the two ethyl groups about this carbon-carbon double bond. Here we can see both the ethyl groups, they lie on opposite sides. To make it more simpler, we can represent or we can imagine this structure is somewhat like this. Here is the double bond. One is above the plane, one is below the plane. If this double bond is representing the sp2 hybridized two carbon atoms. Okay, and now we can compare the identical groups. Are they lying on the same plane? They are not lying in the same plane. They are lying opposite. One is up, one is down. Isn't it? Now, let us, the second structure. Here, if we observe, this is the carbon-carbon double bond, and these are the two functional groups which are attached to these doubly bonded carbon atoms. Here we can see both the functional groups or both the ethyl groups, they are attached to same side of the double bond. To make it simpler, we can represent the same structure like this also. Okay, here we can clearly observe the two functional groups or two similar functional groups or same functional groups, they are lying on same side of the plane, that is above the plane. Here, compare the structures, they are lying opposite. Here we can observe they are lying on the same side. Okay. So what we have learned till now, if same functional groups, they lie on opposite side, we call it as trans. If they lie on the same side, we call it as cis. Now let us see, can we compare the position placement of hydrogens also in such structures? Now let us see. Here we have taken a molecule where the carbon-carbon double bond is attached to two different types of atoms or functional groups. Here it is ethyl group and an atom of hydrogen. Here also we can say there is one atom of hydrogen attached to one side of the double bond and to one side ethyl group. Now, to make it simpler, we can represent the same structure in this orientation also. Here we can clearly observe the two hydrogen atoms. They lie opposite to each other. Now, if we compare this structure, what we can see here, we can simply simplify this structure, or we can represent the same structure or same spatial orientation this way also, where we can clearly say both the hydrogen atoms, they lie in one plane. So which one is called as cis orientation and which one is called as trans orientation? The cis and trans designations included in the nomenclature of alkenes is usually utilized to distinguish between cis and trans isomer. So with this example, what we can conclude, depending on the position of hydrogens also, the way depending on the position of similar functional group, we could determine whether the diastereomer is cis isomer or trans isomer. Here also, based on the orientation of the hydrogen atoms about the doubly bonded carbon atom, we can easily distinguish which one is cis isomer and which one is trans isomer. This isomer is cis isomer and this is trans isomer. Okay. Now let us take another example. Here is the example. That is 2-pentene. What is the peculiarity of the molecule? It is not symmetrical. That is, doubly bonded carbon atom is not associated with similar functional groups on its both sides. Here there is a methyl group, whereas here there is ethyl group. So the molecule is not a symmetrical molecule. In this case, what we will say, this is cis 2-pentene or trans 2-pentene.
Neither cis or nor trans.
No. Here we will see the next thing into consideration. If similar functional groups they lie, if the molecule is symmetrical, in that case, either based on the nature of the functional group or similarity of the functional group, we can decide whether they are cis or trans isomers. Likewise, here, what is the common thing? The carbon here is attached to CH3 group here. There are two double bonds. What about the fourth valence bond? The carbon here
Hydrogen.
is bearing a hydrogen atom. Okay, so where is the hydrogen? It is lying somewhere here. It's not it. And in this way, here it is lying up. So what type of isomer it is? Two hydrogen atoms are common. Very good. Very good. The same thing explained here by showing the invisible hydrogen, which we could not observe here. That hydrogen atoms are highlighted here, and we can say that if there are two hydrogen atoms which are not shown in general in a bond line structure, even though they are not shown, they are there. Now, if we draw these two hydrogens, it becomes more easy and it becomes evident that they can be either easily distinguished into cis or trans, just like the presence of alkyl groups in 3-hexene. So to summarize, we can say as long as there are two identical groups or two identical atoms, they are lying on both carbons, they are lying on the same side of the carbon-carbon double bond, then the alkene can be easily classified either as cis or trans. So it is not only the position of the alkyl group, even with the position of or the orientation of hydrogen atom about the doubly bonded carbon atoms, we can easily distinguish cis and trans isomers from each other. Now, what about non-stereoisomeric alkenes? In order to understand that, now let us see first what are these non-stereoisomeric alkenes. Now, when we talk about these non-stereoisomeric alkenes, one important thing to emphasize here is that the identical groups. Now, here we can see this is the carbon-carbon double bond, and it is representing the plane or sp2 bond. Now, if we see this first carbon of the carbon-carbon double bond, here it is associated with one ethyl group, that is CH2, CH3 on one side, whereas on the other side here there is hydrogen, which is not shown in this bond line structure. However, if we compare this carbon, or if we see the attached functional groups to this second doubly bonded carbon atom, we can see the two functional groups are attached like this. Okay. Now, when two identical groups on the same carbon, two identical groups on same carbon are present, they are called as non-stereoisomeric. Okay. So we can say the most important thing to emphasize about non-stereoisomeric alkenes is about the identical groups. These groups must be on each carbon atom of the double bond. If any of the carbons in the double bond is connected to two identical groups, here you can see two identical groups, then the alkene cannot be cis or trans. That is to say, the alkene cannot be stereoisomeric. For example, this structure we can see the following alkene has two ethyl groups on the right carbon, and we cannot say that there are two ethyl groups on the same or on opposite sides of the double bond. Clear?
Yes, ma'am. Yes, ma'am.
Same limitation applies to terminal alkenes also. That is, if the double bond is placed in the first position, that is in between carbon 1 and 2. Here we can see a terminal alkene, that is alkene bond or double bond is present in the, it is associated with the terminal carbon. Here, if we observe, the carbon atoms are attached to two hydrogens. Here the first carbon is attached to two hydrogens. It is shown here, and here in this bond line structure it is not shown. Okay. If this is the structure, in that case the alkenes cannot be called as cis or trans. तो इसको हम क्या कहेंगे? Non-stereoisomeric. The cis-trans designation is not a comprehensive solution for internal alkenes either. It only works when two identical groups are present on the two carbons of the double bond. To illustrate this, let's consider two isomeric alkenes having four different groups on the double bond. Here we can see four different functional groups. They are attached to the doubly bonded carbon atoms. Okay, and they are, here we can observe all the attached functional groups or atoms, they are shown in different colors. And what they are indicating? They are indicating that all the attachments are different from each other. They are different from each other. Here there is an ethyl group, here a chlorine atom, here hydrogen atom, and here one bromine atom is attached. Now, this structure can also be represented like this. Okay, so if we get examples like this, in that case, can we call it as cis isomers or trans isomers? There is no functional group similarity. Neither alkyl groups bear similarity, nor we can find the presence of hydrogen on both the doubly bonded carbon atoms. So how we are going to decide which one is cis isomer and which one is trans? It is very difficult to differentiate such type of molecule by using this cis-trans mechanism. Since there are no two identical groups, we cannot classify them, these type of molecules, as cis or trans. Here comes the new type of differentiation, that is EZ designation. Okay, instead of cis-trans designation, we can differentiate them by using EZ designation. And what is this EZ designation? That we will see. Now let us, before we go for the EZ and its explanation, let us discuss some of the few exceptional points associated with this cis-trans isomerism. To be specific, the cis-trans isomerism of cycloalkanes. Now, as we have discussed earlier, alkanes, they have got sp3 hybridization. However, when we talk about cycloalkanes, let us talk about cyclohexane or cyclopropane or cyclobutane. When we talk about structures like cycloalkanes, what happens? The origin of cis-trans isomerism is because of the restricted rotation, that is the locked feature of the double bond. And why it is locked? It is locked because there is no free rotation around the double bond. And this in turn means that we cannot switch the orientation of the groups on the double bond. It's not it. There is a free rotation about single bond. And that is the origin of conformers, or when the isomers are created or generated because of free rotation about a sigma bond, then we call those isomers as conformers. Now let us see some example of conformers. The conformers and stereoisomers are described as conformations and configurations. Here we can observe it is sigma carbon-carbon sigma bond. When there is single bond, we call it as sigma bond. When there is double bond, we call it as pi bond. If there is a sigma bond, then there is possibility of free rotation. If we rotate it by 180 degree, what will happen? The chlorine atom, which is facing downward, it will face up, and we can get a structure like this. If both the chlorine atoms are facing opposite, or they are lying opposite to each other, we call that orientation or that conformer as anticonformer, and this conformer as synconformer. These conformers can be summarized as same compound which are having interchangeable functional groups because of the rotation through a sigma bond. Term conformer, I repeat. What are conformers? Conformers are same compounds in which the functional groups or atoms attached to a sigma bond can undergo free rotation. Okay. Now here we can say conformers are same compounds which are where the functional groups are interchangeable through a rotation about a single bond, sigma bond, sorry. Now let us see how these conformers they differ from these stereoisomers. Stereoisomers, when we talk about stereoisomer where there is a pi bond, can we subject this functional group to undergo free rotation? No, because pi bond they are associated with restricted rotation. Double bond is always associated with restricted rotation. So there will be no rotation to get a structure like this. So we can say stereoisomers, they are different compounds in the sense different configurations which are not interchangeable. Conformers are interchangeable, whereas when we talk about stereoisomers, they cannot undergo interchange. These conformations, they represent the same compound and the conversion is possible because of free rotation about sigma bonds. On the other hand, the different arrangement of atoms on the double bond cannot be changed since the atoms are locked due to the double bond. There is no rotation, free rotation about double bond. And that is the configuration of the double bond that cannot be changed, just like R and S configuration of a chiral carbon atom. Therefore, cis trans isomers are called as stereoisomers. Okay. I repeat, the configurations of double bond cannot be changed, just like RS configuration of a chiral carbon atom. Therefore, cis and trans isomers are called as stereoisomers. This comparison brings an interesting question, that is, whether we can ever have cis-trans isomers for a single system with single bonds. If we ask this question, the answer is yes. There are cis-trans isomers for systems with sigma bonds also. Here we can see. Now, if we see these two structures, what you will call this structure? Cis isomer or trans isomer? Both the same.
Cis isomer.
Very good. Here also we can see some different orientation. Two CH3 groups, they are lying opposite. So we will call it as trans isomer. However, here we can notice another thing. All the bonds are called sigma bonds, isn't it? The restricted rotation about the single bond in cyclic systems make it possible to distinguish cis and trans isomers depending on the relative orientation of the groups which are connected to the ring. The ring itself is representing the plane and the functional groups which are attached to the carbon atom of the ring will decide whether the ring structure is having cis isomerism or trans isomerism. The substituents of the cis isomers, they lie on the same side of the ring, whereas in case of trans isomers, they lie on opposite sides of the ring. Okay, the same structure can also be represented like this. Here it is the cyclohexane, and it is associated with two CH3 groups. They are lying on the same side. So they are represented by solid bond on both the carbons. That is, they are both the CH3 groups are lying towards the observer. Now here we can see both the CH3 groups, they are lying opposite to each other. That is, one of the CH3 group is towards the observer and second is away from the observer. Hence we can say this is the cyclohexane. One CH3 group is away from the observer, one CH3 group is towards the observer. And this can be called as, this type of orientation can be called as trans orientation. That is, this is called as cis orientation of 1,2-dimethyl cyclohexane. Here also the same molecule 1,2-dimethyl cyclohexane. However, the difference is that it is cis isomer, whereas this one is trans isomer. Now, sometimes it has been observed that the cis and trans isomerism is also used even if two groups on the ring are not identical. In the earlier example, we saw two cyclohexane at position 1 and 2, two CH3 groups are attached. Here we can see one chloro atom or chloride atom is attached to one carbon, whereas to the second carbon we can say CH3 group is attached or methyl group is attached. Here, and one more thing, here we can see both the both the attachments, they lie towards the observer. Here we can observe the solid bond. Whereas in this case, the same structure, however, the orientation is little bit different. The same cycloalkane, here the methyl group is lying away from the observer, whereas chloride atom is, or chlorine atom is lying towards the observer. Both orientations are different. This is called cis orientation, and this type of orientation is called as trans orientation, even though both the functional groups, they are not same. If they are same, then definitely, if both the groups are same and they lie on the same side, we call them as cis isomer. However, even if they are different based on their difference in the orientation of the functional groups or atoms that are attached to this carbon-carbon sigma bond, we can say whether the molecule is cis isomer or trans isomer. And this is similar to what we discussed about double bonds with different alkyl groups. Now remember, in this case, we focus on the orientation of hydrogens. If both the hydrogens are on the same side, then we call it as cis. And if they are on opposite sides, then we call it as trans isomer. The same example here, the difference is that here the invisible hydrogen is made visible. Chlorine atom at position 1, methyl functional group at position number 2. However, both these attachments, they lie towards the observer, whereas the fourth atom which is attached to this chiral center, that is the hydrogen here and the hydrogen atom over here. If we compare their position, they both are lying away from the observer, whereas here one of the hydrogen atom is towards the observer, whereas the other is away from the observer. If both the hydrogens, they lie on same side, we call it as cis isomer, if the two hydrogen atoms, they lie on opposite sides, we call it as trans isomer. Okay, so with this, all the descriptions of cis-trans type of isomerism is over. So cis-trans isomerism, to summarize, we can say it is not only observed with doubly bonded alkenes only, it is also observed with cycloalkanes. Clear? Now let us see what is EZ nomenclature. Which about this EZ nomenclature I talked in the previous slides also. However, when there is whenever there is it is not possible to distinguish the alkenes by cis trans mechanism, we can distinguish them by EZ nomenclature. What is this EZ nomenclature? The cis-trans approach works only if two identical groups are connected to the double bond. However, if four totally different functional groups are attached to the doubly bonded carbon atom, in that case, we need some more efficient method. When we talk about science, definitely we keep on introducing new concepts. When cis-trans concept was introduced, it made it easier. However, later on it was observed that cis-trans mechanism is okay, but when we come across molecules like this, what will happen? We need something more efficient. So came the new concept, that is EZ concept, or EZ designation is used to differentiate such type of alkenes. Now let us see in the coming slides how this EZ nomenclature is done. Now when we talk about EZ nomenclature, how this EZ configuration is of a double bond is determined. It is determined based on the priorities of the atoms or functional groups which are attached to the double bond. Now you may ask, what is this priorities? Now if at all you recall, when we discussed optical isomerism, I gave you the priority list. It's not it. This priority list is given in all the textbooks. We have to remember that priority list. Once we remember the priority list, we can easily distinguish that which functional group or atom attached to the double bond has got higher priority, which one is having lower priority. Now if we compare this structure, this double bonded carbon-carbon is associated with 4 different atoms or functional groups. Now we have to determine about this plane. There are 2 attachments about this plane. Here there are 2 attachments. Now let us determine the priority among these 2 and the priority among these 2. When we compare the priority of A and B and compare the priority of C and D, we can find that one of them is having higher priority and the other is having lower priority. Now suppose A is having higher priority than B and C is having higher priority than D. In that case, can we say about this doubly bonded carbon atoms, the attachments bearing higher priority, they lie on one side, whereas attachments, that is bonds or atoms or functional groups bearing lower priority, they lie in one plane. To make it easier, we have taken this specific diagram. Here we can see A and C. They are now it is assumed that, let us assume that A and C has got higher priority and B and D has got low priority. So high priority bearing functional groups, they lie on one side of the double bond. Low priority bearing functional groups, they lie on the opposite plane. So it is Z. If this is the situation, then we call it as Z. What is this Z? Z is the representation of high priority groups. They are lying on the same side of the double bond. Now, if the case is like one of these functional groups, that is A is having higher priority whereas B is having lower priority, whereas among C and D, D is having lower priority and C is having high priority. Here if we compare these structures, we can find that here the low priority groups, they are lying opposite, whereas high priority groups are also lying opposite to each other. It is E if the high priority groups are on opposite sides of the double bond. If high priority groups lie on same side, Z. Z is for same, or it has got similarity with same side. Okay, Z, same. Now how this principle works? Here you need to look at each carbon of the double bond separately. This carbon and this carbon. Look at both the carbons separately. And the goal is to first determine which of the two groups on the carbon has got higher priority. Next we have to see the two groups attached to the doubly bonded carbon atoms. Next what we have to do, which of the atoms or functional groups which are attached to the doubly bonded carbon atom, they have got high priority or low priority. The priorities are assigned following the same rule as for R and S configuration. So the priority list is not shown here. We can take the same priority, or we can use the same priority list which was provided during the explanation of RS configuration. Now let us see this example. Here it is an alkene where carbon 1 and carbon 2, they are representing, they are associated with functional groups which are quite different from each other. So what we have to do here, first we have to identify what are the functional groups or atoms which are attached to the doubly bonded carbon atoms. Then what we have to do, then we have to determine the priority. When we talk about priority, which of these atoms has got highest priority? When we talk about priorities, the first priority goes to halogens, and higher the halogen, higher the priority. So if we compare these two, Br has got higher priority. Okay. However, that is here we are going to compare the priority about this carbon-carbon atom only. Here, high priority functional group lies above, and here low priority group is lying below. If we talk about this carbon, here chlorine has got higher priority as compared to ethyl group. Here high priority group is lying below and low priority functional group is lying above. So orientation is same or opposite? Same or opposite?
Opposite, ma'am.
Opposite.
Now let us first focus on the left carbon. Here, first we have to observe the first carbon. Okay. Here we can see an ethyl group and a chlorine atom. Chlorine atom has got high priority as compared to ethyl functional group. So it is denoted as priority 1, priority 2. Next, we will see the next doubly bonded carbon atom. Here we can, if at all, we decide the priority of or compare the priority of these two atoms, we can say bromine has got higher priority, hence priority 1. Hydrogen has got lower priority, hence priority 2. Priority 1, priority 2. Now, if we compare this orientation with this orientation, what we can observe, priority 2 and priority 1, they are lying on one side, whereas here priority 1 and 2, they are lying on one side. That is, high priority bearing or functional groups or atoms having similar priorities, they are lying opposite to each other. It's not it. So to see the structure finally, or the final determination is dependent on whether the high priority groups, they lie on same side or on opposite side of each doubly bonded carbon atom. Since both the chlorine atom and bromine atom, they bear higher priority as compared to this ethyl group or hydrogen atom, we can say high priority bearing groups, they are lying opposite to each other, hence it is E orientation, or for the sake of remembering, we can associate this term E with opposite. Okay. Now, if at all, the same structure would have been like this, that is, both the high priority bearing functional groups lying on the same side and low priority bearing functional groups, they were lying on the same side, then what we would have called it as, we have to call it as Z. The term E and Z has been derived from German language, where E stands for entgegen, that is opposite. For the sake of remembering, we can say E can be associated with opposite, whereas the term Z has been derived from the German word zusammen, which means together. Entgegen means opposite. Zusammen means together. So what is the trick? Z stands for them. They are on the same side. This might be enough to figure out what E means. For the sake of ease, we can associate E with the word opposite. So just like the cis-trans classification, E and Z alkenes are also stereoisomers. Now, what is the main point about this EZ alkenes? Because they are not mirror images, they are called as diastereomers. E and Z alkenes, they cannot be mirror images of each other. Hence, they are called as diastereomers. Now, in this structure, what you will call it as E isomer or Z? High priority?
Yes.
Correct. Now, let us see how the IUPAC nomenclature of these E and Z isomers are done. Okay, or how the EZ configuration is subjected to IUPAC nomenclature. Now, observe this structure. Now, if possible, draw the structure if on your notebook. A carbon-carbon double bond. First, to the first carbon on one side, attach one propyl group. On the other side, attach the Cl atom. Whereas to the second doubly bonded carbon atom. On one side, we can see here it is isopropyl group and here it is ethyl group. All the four attachments are different from each other. So we have to go for EZ differentiation here. Now, if we find out the longest possible chain, now the first rule of IUPAC nomenclature is to find out the longest carbon chain. Okay, so the parent carbon chain over here is a 7-carbon chain. This carbon chain has got double bond at which position? Position 4 or position 3? When we talk about a person, we have to give the position of the lower number. Hence, the alkene is at position number 3. Hence, 3-ene. It is an alkene. To be specific, an alkene of such a nature that the double bond is at its position number 3. Now, what are the attachments? One chlorine atom is there. Where it is attached? It is attached at position number 4, hence 4-chloro. At position number 3, there is ethyl group attached. It is denoted as 3-ethyl. And at position number 2, we have got methyl group. So we can denote it as 2-methyl functional group. The parent alkene chain we have recognized. Now we have recognized all the 3 different functional groups or atoms which are attached to this alkene. The next thing after finding out the parent chain is to identify the functional groups, and then we have to arrange them alphabetically. If we want to arrange these 3 things alphabetically, which one will come first? C, E or M? C will come first. Hence, the naming or IUPAC nomenclature of this compound will start with the term 4-chloro. What is the next thing in the alphabetical sequential list? 3-ethyl. That's 4-chloro, 3-ethyl, followed by 2-methyl. And what alkene it is? As because there are 7 carbons. 7-carbon unit is called as hept. 7 carbons are denoted by the term hept. If at all it is alkane, it is called heptane. If at all it is alkene, it is called as heptene. And to add additional specification to this heptene, the exact position of double bond is specified by the term 3-ene. Okay, now you have to notice one thing. When we talk about IUPAC nomenclature, there lies a dash between the number and the alphabet. The first word starts with a capital letter, whereas rest of the things, they will start with small letters only. Okay, it has been observed that most of the times students, they forget to place this dash. So kindly note that whenever there is a space or there is difference between alphabet and number, it must be separated by a dash. Clear? Don't forget it. Now let us find out the same compound, whether it is E isomer or Z, or whether this configuration is E type of configuration or Z type configuration. How we can find it out? First we have to locate the double bond. Then we have to find out what are the functional groups attached to this double bonded carbon-carbon atom. Here, let us take this carbon. Here, we on one side there is propyl group and on the other side chlorine atom. of these two things, which one is having higher priority? Chlorine atom or propyl group? Definitely, chlorine atom has
Chlorine.
higher priority as compared to propyl group. Now, if at all we compare, now let us compare the priority of these two functional groups. That is, here the isopropyl group and here the ethyl group. Now, if we compare these two, here we can see the carbon atom. Carbon is attached to carbon, and this carbon is in turn attached to other carbons. It is more branched, whereas here the carbon is attached to hydrogen, and CH3. That means this carbon is attached to two hydrogen atoms on two sides, and on one side it is attached to another CH3 group. So this one is having higher priority as compared to this one. जितना ब्रांचिंग होता जाता है, अगर प्रायोरिटी का हम if we remember the priority list, we can easily say here both the carbons, they are having same atomic number. So the priority is determined on the next atom which are attached to the carbon atom. This carbon is attached to two other carbon atoms. Here the carbon atom is attached to two hydrogen atoms and one CH3 group. So here the priority is lesser as compared to this functional group. To summarize what we can say: high priority, low priority, low priority. See here, high priority bearing functional groups, they are lying opposite to each other, hence the term E. And this E is denoted by capital E, and it is placed within a round bracket only. Kindly do not draw any other type of bracket. E isomer or E configuration of this alkene is attached, or term capital E within round bracket is placed at the foremost position to give the correct IUPAC, or to describe the correct IUPAC nomenclature of the given alkene molecule. We'll start with the next slide.